Cure, Connect, Transform. There is no greater embodiment of Mayo Clinic's plans to revolutionize healthcare than the Center for Individualized Medicine. We discover, translate, and apply new individualized medicine advances in patient care. Using genomics and multiomics could lead to vast advancements in knowledge of the causes and risk factors for many diseases. Our researchers and clinicians work together to accelerate discoveries in 4 strategic areas. Simplify and scale. In Genomics in Action, we work to transform the practice by providing the knowledge, tests, and tools needed to make it easier for clinicians to integrate genomics into routine patient care. Individualizing cures with genes, bugs, and drugs. Cutting-edge therapies being developed with high-definition therapeutics are driving precision medicine science beyond diagnostics to provide personalized cures. Connecting data to practice. Our exciting work with Omics Data Platform is centralizing genomic and multiomic data into a single repository, which will allow for the most individualized medical approaches possible. A multi-omics future. In Beyond DNA, we are advancing patient care by using molecular information from the microbiome, epigenome, exposome, and more.
The Center for Individualized Medicine and Mayo Clinic are in a unique position to fuel new discoveries to improve care for patients powered by sophisticated research and artificial intelligence. From providing the best individualized care to addressing the world's most challenging healthcare problems, Mayo researchers here are relentlessly pursuing discoveries that deliver hope and better health to people today and for generations to come. Thank you.
Good afternoon, everybody. My name is Jae Sung, and I'm an assistant professor in the Department of Surgery and Division of Rheumatology at Mayo Clinic, as well as a member of the microbiome program in the Center for Individualized Medicine, or CIM. I would like to welcome you all to the CIM Grand Rounds. The CIM Grand Rounds lecture series is designed to highlight the latest in scientific discovery and innovation, and demonstrate how individualized medicine is being translated into the practice to meet current and future patient needs. Today, we're highlighting the theme of Beyond DNA, which looks to advance patient care by using molecular information from the microbiome, immunome, epigenome, exposome, and more. I'm enormously excited to introduce today's speaker, Professor Michael Snyder. Dr. Snyder is the Stanford B. Asherman Professor, Chair of the Department of Genetics, and Director of Genomics and Personalized Medicine at Stanford University School of Medicine. Dr. Snyder received his PhD at Caltech and carried out postdoctoral training at Stanford University. He was a faculty member in the Department of Molecular, Cellular, and Developmental Biology at Yale University for nearly 2 decades, rising to chair the department before moving his lab to Stanford University in 2009. He is a world-renowned leader in the field of functional genomics and proteomics, a pioneer in self-tracking personal health data through multiomics profiling and wearable sensors, and one of the major principal investigators of ENCODE, which is an NIH-sponsored multi-institutional research project using advanced tools to identify all functional elements in the human genome. At the Stanford Healthcare Innovation Lab, Dr. Snyder runs a team of over 100 scientists doing some really inspiring work that is essentially building the future of healthcare into a system focused on disease prevention. Dr. Snyder was elected a member of the American Academy of Sciences in 2015. And on top of all his scientific accomplishments, he co-founded over a dozen biotech firms in genetics, genomics, and precision medicine based on personalized big data, all with the main aim of keeping people healthy. Today, Dr. Snyder will be speaking on new advances in health monitoring, describing how his lab's work during the past few years is radically transforming healthcare with deep multi-ohm molecular measurements and wearable technologies. More specifically, he will summarize his recent findings using those technologies, including the concept of agiotypes, how wearables can detect COVID-19 and other infectious diseases, and remote sampling for health monitoring. For the audience, we'll have a Q&A session at the end of Dr. Snyder's talk. So please feel free to use the Q&A in Zoom to submit your questions. And for those who have not yet claimed continuing education credit, a message will be sent out in the chat shortly. Michael, thank you so much for accepting our invitation. The floor is now yours.
Okay, well, thanks, Jay Young. It's really great to be here and present some of our latest work. In fact, one of the topics I'll present just came out today, Nature Biomedical Engineering. So as Jay Young said, our shtick is to try and use big data to transform healthcare. And let's get started here on the sharing. All right. Great. So I would argue our healthcare system, I'm sure many of you would agree, leaves much to be desired. And there are many ways in which we can probably improve healthcare, but these are some of the elements that particularly bother me. We are very focused on treating people ill. As such, medicine today is very reactive. We measure very few things, especially when people are healthy. Also, when you're healthy, you don't get measured very often. And one particular problem is nearly every decision about your health is based on population-based measurements. Instead, I think we would all agree it's better to keep people healthy, be very proactive. We can certainly measure many, many more things than we currently do. The frequency, I would argue, should depend on what you're at risk for and what your personal trajectories look like. And lastly, we should be doing more individual-based medicine than we currently are doing. You know, we do some, but one could argue we can do this much, much better. And the net result is we should be very focused on precision health rather than precision medicine. So to illustrate this point, I like to show the slide here, which is your oral temperature when you put a thermometer in your mouth. We've told since you were little, it's 98.6. Yet if you actually look at the studies that are out there, this is a very typical one. I say I lost the credit here, but basically from a study of 3,000 people, the median temperature is 97.5. It's not 98.6. And mine personally is 97.3. It's actually been dropping over the last 8 years. I think more importantly, though, is there's a spread. You can see this is a 25th quartile, 94.6. This is a 75th quartile, 99.1. And what that means in today's world is that if your normal healthy baseline is here, 94.6, you go to a physician today and they measure you at 98.6, they'll tell you you're normal, everything's healthy. But if you're up 4 degrees over your baseline, I guarantee you're not healthy. And so that's a big theme of our work. Know what your healthy baseline is so you can detect a shift and catch disease at its earliest onset. So we're in a world now where I think we can start dissecting, things that impact your health. So your health is influenced obviously by your DNA and your genome. A lot of emphasis is placed there. But we know intuitively all these other things impact your health. Exercise, food you eat, stress, various exposures all impact your health. And we're in a world where we can now quantify this. So you can quantify exercise through activity monitors, food by logging, that one's still clunky, stress indirectly, and other things. We're actually ourselves working on a lot of technologies, especially environmental exposures. And so we can quantify many of these parameters. But equally importantly, we can quantify the impact of these parameters by doing deep molecular profiles on people. And so a number of years ago, we set up something we call the Personalomics Profiling project, where in addition to sequencing people's genome, we actually made many other measurements, mostly out of blood. So we'll follow their DNA methylome, transcriptome, proteome out of peripheral blood monocyte cells, so immune cells, cytokines and proteins out of plasma, metabolites, lipids also out of plasma. We do follow the microbiome. We do gut, nasal, and more recently, we just completed analysis of tongue and skin. That turns out to be quite interesting. And these clearly impact your health. This is on top of deep clinical tests, questionnaires, stress echocardiograms. These are more advanced tests, and a lot of glucose control tests, because it turns out I'm type 2 diabetes. We'll talk about that in a minute. And so we do oral glucose tolerance tests, insulin resistance. And then about 8 years ago, maybe a little longer, before we started getting involved in wearables, as biosensors, and so added that on as well. So one aspect of all this is to collect deep data on people. And the other aspect is to do it longitudinally. So we'll sample people every 3 months while they're healthy. And then if an adverse event comes along, say a viral infection, we'll take typically 5 to 7 additional samplings. And so we've been running this on a smallish cohort, about 109 people. Some are now dropping out and we're adding others on. And we've been running the study for, well, I'm one of the participants, 13 years on me. It's almost 10 on the rest. So you might say, why are we doing all this? And the answer is we're trying to understand what it means to be healthy. What does a healthy profile look like? How does it change over time? How does it differ between different people? What happens at the earliest times of illnesses like a viral infection? And lastly, can these advanced technologies like genome sequencing, these omics technologies, can they be better used to manage people's health? And with regards to the last question, omics technologies and health, we actually discovered that from the smallest cohort, nearly half the people learned something important about their health. 49, about 47 people, sent in 2 issues, were found, and it spanned a wide range of areas: hem-oncology, cardiovascular disease, metabolic, so on and so forth. So in each of these areas, people learned something pretty important about their health, all in advance of symptoms. So that was what's important about this. We could, by doing these deep data collections, we could see when things were off in advance of symptoms. And it was no one technology that did it. Sometimes, we'll talk about this more in a minute, sometimes it was genome sequencing, sometimes it was imaging. And some of these were a big deal, like we did catch someone's early lymphoma, 2 people with precancers, MGUS, smoldering myeloma, 2 people with serious heart issues, one from genome sequencing and one from wearables, and so on and so forth. So again, all these were found pre-symptomatically. Now, the way we like to think about it is that the way we do medicine these days is taking 8 pieces of a jigsaw puzzle to try and figure out what the picture on that 1,000-piece puzzle is. We're actually trying to take more like 600 pieces where we'll get a much, much clearer picture of people's health. That's really the idea. And so again, quite a bit was found out. So just to give you a few examples, genome sequencing was powerful. 12 people, actually 13 if you count my polygenic risk score, and actually 14 for one of the pharmacogenomic variants. But 12 people from the first 70, it's a little bit higher now, learned something pretty important just from their genome sequence. And these are what we call Mendelian mutations. These are single gene mutations that by themselves with a high incidence can lead to disease. And of course, the most famous one are the BRCA mutations. So we did have an individual with a BRCA1 mutation. That person was an orphan, and learned that then they were at risk for breast cancer. And so again, we found a number of these. It turns out 3 of them actually wound up having impact on disease, meaning this individual here, after we sequenced his genome, we saw that he had a mutation in cardiomyopathy gene. And it turns out his father died of a heart attack at around the same time. Aunt had one in her 50s. And then he did a stress echo. And as a consequence of this genome sequencing, sure enough, he's got a heart defect. He's on drugs now. So the point out of all this is that we could actually uncover this from genome sequencing in advance of symptoms. There's another individual, who had a mutation in this gene, puts them at high risk for certain kinds of cancers. They did a whole body MRI file, had early thyroid cancer that was removed. They were able to keep their thyroid, didn't need thyroid replacement therapy. And the net result is, again, that was all caught early. And both these folks were quite young, actually. So it was a big deal. I forgot to say that the cohort as a whole was 53.4 when they entered. So that's probably why we found so many. It is an older cohort, but it's a widespread. So we did have young folks, like one of these here. 2X means there were 2 individuals. Even some had mutations, APC mutation pathway. There were 9 individuals coming in the study thought to be type 2 diabetic. And one of them actually turns out that mutation in this gene, they're MODY diabetic, which is a different form. Many of you, I'm sure, know. And the way you treat a MODY is often different from that of a type 2 diabetic. So they've actually got on better medication now. They've been on suboptimal one for years. So net result, again, is that even though it's a minority of folks who learned something valuable from their genome sequence, if you're one of these, it's a pretty big deal, like the BRCA person and so on and so forth, the heart person. So we think genome sequencing can be quite powerful. Many of you may know that most forms of disease, when you think about genetics, they're really not due to these single gene mutations like BRCA and stuff. They're actually due to complex disease. And most complex disease, you may or may not know, it's thought to be due to these common variants, these common changes in your DNA that are thought to be a very small effect. And people are unfortunate, get a lot of, the bad variants for these things, and type 2 diabetes, rheumatoid arthritis, all these things that they're the most common forms are the complex disease. And so what people are doing, so you can interpret these single gene mutations the way I just described. You see a mutation, if it's a known pathogenic variant, you kind of can then coach folks. For complex disease, what people are doing, I forgot to say, the analogy is that this is like clobbering a gene with a sledgehammer, and then therefore leading to disease. And this is more like, death by 1,000 cuts. Lots of little changes, a small effect, basically cause the problem. So the way people are dealing with complex disease these days is using something called a polygenic risk score. So we're going to do a touch of genetics here. And in polygenic risk scores, what people do is they sum over these variants. Well, they have an increased odds or a decreased odds of causing disease. And these days, people will sum over thousands, if not millions of variants. Here's a paper from a few years ago, looking at these 5, again, complex diseases. They'll sum over millions of variants. And this sort of works. It works for the top couple percent. So if you're in the top 2 or 3%, you can actually get pretty good predictive value. But if you're below that, forget it. It doesn't work that well. And we think that there's 2 reasons for this. One is the way they do this is by linear summation, and genetics isn't linear. For any of you who are geneticists, you probably know that 1 + 1 is rarely 2. There's interactions like 1 + 2 mutations in the same pathway. 1 + 1 there would be 1, because you won't get additive scores. And if you're different pathways, sometimes 1 + 1 can be 15. That can be very additive, but more than additive effects. And so that's all missed. And the other thing that's missed are all the rare variants that contribute to this. So this only assumes these common variants. So we came up with some new methods for analyzing genomes that have been quite powerful. And I'll tell you about one of them in a minute. They both use machine learning, which I'm sure you appreciate is really transforming a lot of different fields. When we first applied this to looking at a disease, in this case, I'm going to give you the rare variant example. I'll show you, I'll just summarize a common one in a minute. But we looked at a disease called abdominal aortic aneurysm. You may know that, but this is actually a big killer of people over 65. It's actually the 10th leading cause of death. And unfortunately, the way it's discovered is that when people's aorta bursts, then they die 90% of the time. So that's a terrible way to know you're at risk for the disease, obviously. It's known to be highly heritable, 70% heritability. And there are some markers associated with this. So we wanted to see if we could try some new machine learning methods to get at the genetic basis of an abdominal aortic aneurysm. And what we did was something that would just totally, and it did at the time when we published, it was totally mind boggles GWAS folks. So if you're a GWAS person, you're probably going to say, hey, there's no way this is going to work. And it works great. But the idea is the following. We sequence, we only have we got 401 great genomic sequences, 268 cases, 133 controls. We did do whole genome sequencing, but we're really just calling pathogenic variants inside the gene. So we actually did a decent depth of sequencing as well, 50x. And then we did this machine learning where you basically say, give me all the pathogenic mutations. Give me the genes that are rich with pathogenic mutations in cases relative to controls. We also, you can mix in other features while we mix in the limited amount of EHR data. And once you know it, at the end of the day, we came up with 60 genes. There had only been 3 genes known before, and even those were kind of sketchy. We found 60 genes underlying, in this case, that were hyper-rich for pathogenic mutations relative to control. You do, for the aficionados, 10-fold cross-validation and can show all this. And it's like a 10 to the minus 13 p-value is what comes out of this. And then we went on, I won't have time to show you this, but we could show the pathways that the 60 genes are involved in. They're exactly what you expect. Blood circulation, blood pressure, cardiomyopathy, aneurysm. We also looked at mouse models for AAA, and sure enough, the paralogs of this, they're all involved in cardio and these aneurysm kinds of functions. And lastly, we could show the genes are misexpressed in both AA patients and in the mouse models. I won't show you all that data, but there's a lot of validation around this to show that these 60 genes were, in fact, the right genes. And then we could even go on and build a predictive model that actually we think could be used clinically. So we're trying to test this out. But basically, the genome alone actually has reasonable predictive value, 0.7. Electronic health records can be valuable, especially later in life, because they'll show high cholesterol and can be predictive. But the two together are even better. I know that looks modest, but actually that's a pretty big jump in sensitivity. So at the end of the day, we now have a predictive model, and I know as we add more and more data, we'll get even better. And so that's what we're trying to do. And the model's as good as certainly the coronary artery disease models that are out there now from Framingham and the more recent versions of that. So we think this is going to be quite powerful. We've now gone on to actually do machine learning in other ways for two other diseases, some ALS, which many of you know, it's also known to be highly heritable, 61%, and severe COVID. So young people get COVID, can wind up in the hospital. And that's thought to be due to genetics. Here we did a little bit different approach. We published this, so feel free to, I can share the slides. You can look this stuff up. But basically, there were 7 genes. The biggest study out there ever done, they found 7 genes. And with our machine learning, we can actually search the open chromatin regions and look for enrichments in GWAS signals in these open chromatin regions relative to control. So for that case, we focus on motor neurons. At the end of the day, we identified 690 genes. So we basically 100 times more genes. And we went on and validated several of these. And in fact, we validated all of them by many methods, misexpression, all the same stuff I showed you before. Very, very powerful. And excuse me, we knocked out one of them, TANK1, which also turns out has a Mendelian form that was discovered as we were doing this. And sure enough, that has all the phenotypes of an ALS. So the net result out of all this is that these methods are very, very powerful. Same with severe COVID. This case, we use open chromatin regions from single cell data and can look for enrichments across all 19 cell types. We got over 1,000 genes involved. We can explain it's a whopping 77% of the heritability. That's like unheard of. It's amazing. We can really pin down the genetic basis of severe COVID. That's because it's a giant GWAS study. So again, the net result is that, and that's true for everything we're testing now, this is just a very, very big enrichment in finding genes. Now, we don't yet have predictive models for these. We're working on that. I'm pretty confident we can get there. Happy to talk about that later. But the net result is these machine learning methods, they work for 2 reasons. One is we restricted the search space for open chromatin. Sorry if it's a little jargony for some of you. And the other is the fact that we're doing machine learning, which is nonlinear, and the two are quite powerful. All right, so back to the cohort. Well, one of the things we did learn is that other technologies were valuable too. Imaging was valuable. We caught some of the early lymphoma because they had a large spleen, and several blood markers that were up. We caught some with this pre-cancer called MGUS. Many of you may know it. It's when people have too much IgM and too many IgM producing cells. Now, this one did come out from basically the cohort analysis, meaning we had one individual, a pretty young person, had just way more IgM than everybody else. There's 1,000 samples. 2 samples were just way higher than everyone else's. So it was quite revealing, and in follow-up said, yep, got this MGUS. And so get monitored more frequently as a consequence. We have found other discoveries as well. Just tell you one last example, which is type 2 diabetes. This is one that interests me a lot because I mentioned I am type 2 diabetic. And so basically coming into the study, I mentioned 9 people are thought to be type 2 diabetic. They're the ones in blue here. Well, 2 people are diabetic who didn't know it. That does happen. And a lot of people are pre-diabetic who didn't know it. That's very, very common. And then we're very interested in how do people become type 2 diabetic? Do they just gradually get there? Does something trigger it? How's that work? And in the first 4 years, 9 other individuals became diabetic, and a few others had diabetic range values, weren't officially diagnosed, and others became pre-diabetic. So with regards to how do people become diabetic, the answer is of the 9 who clinically were diagnosed, 7 of them just gradually got there. So here's 2 examples here. Glucose is in orange, hemoglobin A1C is in gray. And you can see this person gradually just went up on both of these, and then actually an intervention, talking about that later, brought them back down somewhat. Here's a person who got there from oral glucose tolerance test, was in range, although nearly there, and then just got there gradually. But of the 7 that did this, so 7 gradually got there, 2 actually gained weight, and that's a classical way to become type 2 diabetic. The other 5 did not gain weight. Now, for 2 other people, it actually looks like something triggered it, and I'm one of the 2. This is me in 8 years of data. Like I say, I have 13 years now. In my case, I first became diabetic after a nasty viral infection. I was actually getting measured for an insulin resistance test, and surprisingly, my fasting glucose was high, and I had elevated hemoglobin A1C, and got tested a few weeks later, and sure enough, was basically crossed the threshold, officially classified as diabetic. And in my case, then I did a lifestyle intervention. I basically cut out all sugars. I've been doubled my biking. I've been biking some, but basically cut out all the excuses. And I started running, and I gradually brought it down. Now, you may say, well, that's a little bump, but that's actually about 10 months to a year before I brought it down to normal range values. And so I was able to get it under control by lifestyle changes. Now, it was running so boringly low that I stopped looking at it. I did the perfect control experiments. I wasn't looking at the data. And someone else noticed here a few years later that my hemoglobin A1C was back up high again. I said, oh, I was surprised. I went back and looked. I forgot to say, so this first one came after respiratory syncytial virus infection, which these days is running rampant. It's kind of interesting now, catching a nasty reputation. I got pretty sick when I had this. This was back in 2011, or was it 2012? 2011, yeah. So, and when it spiked up again, so we went back and retrospectively looked, and sure enough, it spiked up again. And 2 things happened. I stopped running, and I also got a second viral infection. So it's not clear what triggered this, but nonetheless, it's pretty clear I have a glucose dysregulation. I forgot to say it was predictive from my genome, from my polygenic risk score. And so I'm at the extreme end. That's why we could predict it. Now, I did start running again. I brought it down. I never got it all the way down to baseline levels, but I did get it pretty far down. And then I was running in about as much as my schedule could handle, 4 days a week, 4 to 5 miles a run. And gradually, both my hemoglobin A1C and fasting glucose kept going up and up. And so I switched from running to weightlifting. You can't tell, but I lift weights. And the idea is that weightlifting and muscle mass is better for glucose homeostasis. And so I was hoping that would bring it under control. That failed. I did gain 10 pounds. I measured myself of muscle mass. I do whole body MRIs. But unfortunately, I just kept going up and up. And that was a little disappointing to me, to say the least. So finally, I bit the bullet and did what all my friends, my MD friends, were telling me, Mike, you have to take metformin. So I did that. And guess what? I'm a non-responder. I just kept going up and up. I went all the way to 7.5. It's not shown here. And so what was the solution for me? Well, the answer is more data. And it's pretty clear at the end of the day. First of all, I obviously have a glucose dysregulation defect. I actually make insulin fine, and it turns out I'm insulin sensitive. So my cells respond. So what's wrong with me? Well, I actually don't secrete insulin from the pancreas. I have a very slow glucose disposition. And we went on to deduce. It turns out I'm a good responder to rapidolide, which is a prandin derivative that promotes release in the pancreas. And then that works. And then I switched to an SGLT2 inhibitor because they have cardiovascular protective. The reasons that's valuable for me, that also worked. So then, and more recently though, they worked but didn't get me low enough. So, and it kept creeping up again. So more recently, I went on the GLP-1s, and that's like 2 weeks ago. And that seems to really work amazingly well. So the net result out of all this is what we call precision diabetes. By better knowing what was wrong with me, we could prescribe the right drugs. And a lot of this stuff is very predictable from the data if we were to only run the tests for folks out there. So I honestly think we can use this kind of information. I know what we're showing is a research project, but we can easily adapt this to the clinic. All right, one of the most important things we learned from all of this is that people have personal profiles and they're actually very stable, and they're also very, very personal, they're individualized. They're different from one person to the next. So as an example, I'm showing you data from 12 people and who had at least 10 or more healthy visits. I have 6 years of data. I'm the light blue one here. And no matter what OM or test you use, people cluster according to themselves. Like say, I have 6 years of data, all my spots, I'm the light blue one, cluster together. Here's the red person, light blue, purple, so on and so forth. People do cluster on top of each other. Even this transcriptome will do the same. And so, again, these profiles they're personal, they're stable. And even if you undergo a perturbation like a viral infection, in this case, people ran to the VO2 max. They actually shift their profile. Half your molecules will change if you run to your VO2 max. But at the end of the day, you will still look more like you than the person sitting next to you. So I did the experiment twice. On these two, one's the dark brown, the other's the yellow. And mine, a year apart, mine jump right on top of each other. And then here's another person, so on. So the point is that the differences between individuals is greater than the difference between a perturbed state, like running to the VO2 max, or even getting a viral infection. It's not 100%, but it's mostly there that you do shift when you get these perturbations, but you still look more like you than anyone else. That's a really, really important concept. Because if I want to tell the difference between a healthy you and a sick you, if I'm wondering if you're sick, it's very hard to do that by taking sick measurements and comparing it to just a lot of other healthy people. But if you compare it to their baseline, it's a very easy thing to detect. So we're big proponents, get your healthy baseline now and find that shift. So the other thing we can do, I mentioned most molecules are pretty stable. Well, there are some that do shift over time. And so what we've discovered, we look for patterns of things that might shift. We found that actually there are seasonal patterns in the data. That turns out to be very, very interesting. But we also notice these changes over time. There's 600 molecules of microbes, microbiome microbes, that will shift over time. And what we discovered is that people actually age differently. The different molecules that do shift differently in different people. So this is me, about 4 years of data. Here's another person with about 3 years of data, or less. We think with as few as 5 measurements, we can tell how you're aging. And what we discovered is everyone's aging differently. So this is me, my coagulation, metabolic, other pathways go up with time. And that's a very typical pattern, actually. But you look at this person, their top pathway that's changing over time is their cardiac hypertrophic signaling pathway, their cardio. So this person's a cardio ager. And so it's so we think their heart's off. Basically, we later learned they're stage 2 hypertensive, and that sort of fits with this idea of cardiovascular functions off. So what we discovered then is, we had 43 people with enough data. We said, how many patterns are there in the data? And at the end of the day, there were 4 patterns. We call these ageotypes, 4 aging patterns: kidney, liver, metabolic, and immune. So each column's a different person. Each row is a different ageotype. And the folks over here, these 4 are aging in all 4 categories. This person's aging in 3 of the 4, not much of a kidney ager, but aging the other ones. This person's kidney, this is kidney and metabolic. I'm in the middle. I think this could be me, actually, probably that one. Not much of an immune ager, but age more on the other things. So end of the day, we can see how people age. It turns out the information is clinically actionable. There are markers associated with these ageotypes. I'll just show you 2 examples. Here's hemoglobin A1C, this diabetes marker. The group as a whole goes up, and 4 individuals, the dark ones, that's statistically significant, they go up as well. But interestingly, 4 other people actually went down. And this is a simple one to interpret. They did an intervention. So 2 lost weight, 2 went on a diet, 1 started running. And so that undoubtedly explains this shift. There's another one with creatinine. And here, what's interesting here is a marker of kidney function. The group as a whole goes up as expected, but 10 individuals went down. They're statistically significant, and 8 of the 10 are on statins. So there might be something interesting there about statins. There still are many things to be desired about how we can do healthcare better. And I'm going to talk about now remote monitoring. If you think about what we do these days, people, they travel to a doctor's office. They go to the office, looks a lot like one of 4 years ago. Mayo ones are nicer. I've been there. But nonetheless, many of the technologies are the same. A few of them have shifted, a few more gadgets, but they haven't changed that much. Then they draw lots of blood from you. Sometimes it hurts. And then they run a few measurements I mentioned before to see, what your profile looks like. So we're believers that this whole thing can become a lot more done at home. And so we've been doing a ton with wearables, which I hope to get to in a minute. And also most recently, the paper that came out today talks about our work doing microsampling from tiny drops of blood, the kind of thing you learned about from Theranos. And here the idea is that you take a tiny drop of blood. We're using Matrix and another thing called Tasso devices. So we spent a lot of time, we spent 6 years trying to find the right technology. And so in the end, we thought these were very good devices for collection. You FedEx a sample to our lab, the lab process it, and then we can do these deep profiles, lipidomics, metabolomics, proteomics, and very targeted assays as well. So again, the idea is sampling's at home, and then the analysis kit's reported back. And so we first showed that most of the analytes we measured are stable. That was, again, part of the whole testing process. We put samples, we took blood from 2 people, one of them, put them at 3 different temperatures, along with storing them at -80 right away, and put them at different lengths of time. Turns out this is a pretty massive experiment to look at stability across time and temperature. And then we also, and we scored this. And at the end result was that this duration, this is temperature, this is something called an interaction term, a combination of the two. And nearly all the analytes are stable to time and temperature. So of 128 proteins, most are stable. A lot of these are targeted assays. Same is true for metabolites. Most are quite stable. And lipids, a lot are off. Half of them are off. And we don't think that's a big deal because you can actually just work quicker. And in the long run, you could actually measure exactly what's going on. So the net result is that this assay generally works for most analytes. And then we can also correlate, remember, a microsample is whole blood, and we tend to compare this with plasma. So we did do that from the exact same person the same day, the microsample and the blood. And sure enough, there's a pretty good correlation in these analytes between whole blood and plasma. And again, they're different assays. For lipids, it's almost perfect correlation. For the metabolomics, it's more off, but well, most of them are spot on line, but some of them are a little bit more different. So anyway, and we can correct for that if we want. So we went on to use this in 2 different ways. I'll tell you about 2 studies, probably going a little fast. I hope it's not too fast, but it's kind of fun, these studies. I'll summarize the main points. We decided to try this out by running what we call a shake study. Now, you probably know that everybody responds differently to different nutrients, but exactly how, and it isn't so clear. So we had 32 people. We wound up getting a full data set from 28. Sometimes people miss a spot. What they did was they drank this Ensure shake, which is a complex mixture. And then we measured them at baseline, 30, 60 minutes, 120, and 240 after drinking the shake. And as I say, it's a very complex mixture. And then we can go in and see what the response is. And it turns out, 221 molecules did shift after drinking a shake. And there are different profiles. Metabolites show up in the blood early, lipids a little bit later. Acylcarnitines go down when you bring in all these other exogenous nucleus. So that's the third. There are 3 categories of molecules here. And if you look at some of the key molecules, this is some of the things involved in insulin response. You can see we can measure all these things from this tiny little, they're 10 microliter microsamples. And one of the cool unexpected results was that, leptin actually went down, and some of these inflammatory things that actually dropped with the Ensure shake. I'll come back to that in a minute. Turns out that, I'll come back in a minute. So one of the things we discovered is that different people have different responses to, the shake. And so this is the carbohydrate response. Generally, the class of molecules behaves the same. for any given individual. So the carbs, for example, all behave the same, amino acids the same, so on. So here's an individual, look at their time course. These varies 30 minutes, 60 minutes, 220. This individual, their carbohydrates drop after drinking the shake. Same with this one over here, pretty dramatically, actually. Here's a third one over here. Then you have other individuals like this one, their stuff skyrockets, this one, that one, so on and so forth. So different people are responding very, very differently to the shake. And we can go on and score all this. This is, for simplicity, we just picked 6 categories. Gray is the average score of the group. And then I'll just show you a few examples. This person over here, they're elevated for inflammatory markers, inflammatory proteins. They're decreased, they're increased for free fatty acids, but decreased for amino acids. And then this person's quite the opposite. They're actually anti-inflammatory. There are a lot of inflammation markers drop in response to the shake. Free fatty acids go up. Let's see, this one goes up to amino acids. So people are responding. We teased out 5 categories of people, and these are just some examples. Just look at this guy. Their amino acids go way, way down after drinking the shake, and inflammation up. So again, people are responding very differently. And we think this is a big deal because, if 10% of people have inflammatory bowel syndrome, and if you can actually identify that and who they are, you could actually modulate what they should be eating and drinking. And so I think we can start pairing diets to their effects on inflammation, other markers based on this stuff. The other thing we did was we wanted to see if we could do detailed profiles on people. So we picked a person, turns out it's me. You can tell I didn't make the slide because it's my picture, but we did very, very detailed profiling on me for just over a week, a little over 7 days. We collected 98 samples, so roughly almost 1 an hour across all waking hours, including one in the middle of the night. So we're doing this dense sampling and then dense profiling for proteomics, metabolomics, lipidomics, targeted assays. I was also wearing a smartwatch at the time and a continuous glucose monitor doing food logging. So you can really relate all these variables. And the net result is you can start teasing out the pattern. So here's glucose monitoring, here's heart rate. I'm type 2 diabetic, remember, so I will spike. Steps, food log, so on and so forth. These are the molecules that are showing a shift. Certainly the majority of them do show some sort of shift. And so here's a sanity check. This just shows that basically I drink a shake every morning, 2 components in the shake. They go up, come down. That makes a lot of sense. Take a baby aspirin, used to take it every day. I missed a few days in this particular sampling period. So you can see my baby aspirin there, but now you can actually determine its kinetics, its metabolism kinetics. And so one of the things we learned that's kind of fun is that even though I stopped drinking caffeine and coffee by noon, and even though I did that, I still had a negative correlation between caffeine levels and sleep. And so the net result is now I drink less and I try and stop before noon if possible. So I better get my next sip soon. And we'll see if that actually improves things. I should have done that. We found lots of molecules were behind, so I'll skip over this, but we find lots of molecules with circadian pattern. Some of them we think are associated with food like this one, but others probably are not, or don't seem to be based on what folks ate. So there's some intrinsic, lot of circadian molecules here. We're very interested in causal relationships between activity and biochemistry and between biochemical markers as well. And so we invented a method to do time lag correlation. So some events, they may not happen right on top of each other. Step A happens and then that causes step B. So you get these lagged correlations, if you will, between events and say biochemistry. And so just to illustrate this point, you can look for a correlation between steps and heart rate, right? So steps, we know, increases your heart rate. And so we could do that. We basically took steps and heart rate and did this lagged correlation association analysis. And the net result is there's a 1-minute lag between steps and increases in heart rate. And that, pretty much makes sense. So get a fairly rapid increase. And then we could do that for all the biochemical markers, all the physical activity. And what we wound up finding is there's thousands of associations between heart rate, steps, and CGM, or at least over 1,000. Yeah, I guess 1,200. Sorry, I exaggerated slightly here. And these are some of the top ones. CGM, that's glucose levels, heart rate, steps. These are metabolites, lipids, cytokines that correlate. But it's kind of interesting that the cytokines, the steps and heart rate show a lot of overlap. That's not surprising, but they also have differences because there are certain things that trigger your heart rate that won't trigger, that aren't associated with steps. Yeah, and then we can see the pathways that change. This is just doing the pathways that correlate with heart rate. So caffeine is number one. There's a nutrient signaling pathway and so on and so forth. Some of these don't, I don't fully understand them. Maybe some of you do. Cushing syndrome, correlates with increased heart rate, whatever that pathway is. So we went on and we've looked at the glucose one in detail, and I'll just jump to the chase. We've looked for some of these time-like correlations. So here's a nice one between, say, CGM and your insulin C-peptide. And sure enough, there is an association here. Your C-peptide comes up about 10 minutes, it turns out, after glucose levels rise. So that's my kinetics of how quickly my insulin's kind of taking action. My guess is because it is taking action a little bit suppressed in the magnitude because of the other data I mentioned. The other discoveries we had is this protein involved in Alzheimer's actually associates with glucose levels as well. In this case, this precedes it. This is stress. It's thought to be involved in stress. Precedes it by 55 minutes. So one could envision trying to decrease this I don't know if this is true, but try to decrease this for preventing Alzheimer's, and you would have a glucose marker as a way of doing that. So that would be of interest. We find all kinds of new associations. Here's one between TNF beta, which is involved in autoimmune and other immune functions with glucose levels as well. They're on top of each other. We can't distinguish cause and effect. Might be triggered by the same thing. So lastly, I say I got behind. Maybe I'll take a few minutes just to summarize, as we're doing a ton with wearables because we think this is really the future. It's inexpensive, can get out to the planet. So many of you may know we're very big on this. And so I normally wear 4 smartwatches. If you look up, I've got 3 on now. One of them, the band broke, so we're down 1 watch. But we basically are using these for health monitoring. So we got involved in this pretty early on, back when they were just being used as fitness trackers. They're powerful because they make hundreds of thousands of measurements on people every day. The good device, the better device, could even make 2.5 million measurements on people. And they'll track all kinds of things: heart rate variability, which is pretty accurate, respiration, SpO2, and blood pressure are not accurate. It depends on the devices. Some don't even have these things. Some have them accurate, some less accurate. Well, not the SpO2 and blood pressure, not accurate on anything. But the deltas are fine. You can see shifts. Skin temperature very much depends on the quality of the watch. And so I'll just summarize. We discovered early on I could tell my Lyme disease because my resting heart rate jumped up and my blood oxygen dropped prior to symptoms. And I spotted this on an airplane. It was abnormally low for the blood oxygen, abnormally high for the heart rate. I later learned my skin temperature was up. It wasn't just on the airplane, it was after I landed just as well. Went on to show that I had 2 years of data. So every time I had an illness, here's a Lyme case, but I had respiratory viral infection here as well, we later learned. This one's an asymptomatic case. And every single time I was ill, I had high resting heart rate, high skin temperature. So we went on to write an algorithm for all this. And we discovered that basically you could actually see there's a delta plot, if you will, the shift up in resting heart rate in advance of symptoms. And it worked on me. These are the 4 times I was ill. And it worked on 3 other people, one of whom got sick twice. Every single time we could see the shift up in resting heart rate prior to symptom onset. So then, we published this in 2017, made a big splash. It was nice. And then we were basically improving the algorithms. And of course, along comes COVID. And with all its waves, we're still getting tons every day. People stop reporting it, but it's at least, somewhere between 60 and 100,000 based on reported measurements, probably way more. And the way you do it now is with temperature, which is a terrible measure of COVID. A lot of people don't get fevers at all. And we think resting heart rate is one of the strongest measures. So anyway, we quickly launched a study to see if we could detect COVID with a smartwatch, simple commercial grade smartwatch. Right away, we got 5,300 people. And this is 32 of them were wearing a Fitbit while they got COVID. This is all retrospective now. And these 32 people had a diagnosis day and a symptom day. And what we discovered is that there's our first person. You can see here their diagnosis day, here the symptom day, but their heart rate jumped up 9.5 days prior to symptom onset. You can't miss a standard deviation plot. And I'll show you in a minute, we have a real-time detection algorithm for being able to detect this. It builds on people's baseline, looks for these shifts. And so in our initial study, we showed that of the 32 people, 26, so 80%, we could tell they had COVID in advance or at symptom onset from a simple smartwatch. And the median was 4 days, pretty good actually for picking up COVID. And yeah, we then built this infrastructure, cloud-based infrastructure. We can run this on millions and millions of people. And the idea is the following: we again, based on your baseline, we'll look for these shifts. It's an anomaly detection algorithm. And the latest one, it's a lightweight one, so we can do it at scale without breaking the bank. And here's one of our first cases again. Here's an individual who, this is their symptom day. Here is when they were diagnosed, but they were getting red alerts. This is a real-time red alerting system. You actually, though, have to click on the phone. We don't yet have it set up so it sends out the alert to you automatically, like the Amber Alert or something. That is the goal. But right now, you have to click on it. Most days are green. We have it set so about every 6 weeks, an alarm goes off. Survey said that's a good number. And basically, you can see this person sick, red alert day, red alert, 3 red alerts, and then symptoms here, diagnosis there. So it turns out it works for asymptomatic cases as well, 14 of 18 asymptomatic cases. We can see the shift. I guess I left that slide out. I want to emphasize it's not specific for COVID. Workplace stress is number one. Intense exercise, all these things can trigger red alerts, including other infections. So we're trying to get more data. I can tell it worked on me. I got sick April 10th last year. Those are real dates for me. This is my alerting. So I had a particular family stress. And again, most days are green, but I did have these days where there was a particular event I had to travel up, so that's trigger. Now I was getting ready for a trip in New York City. It turns out I woke up, I was a little bit congested. So I did, my antigen test, that's over here. I was negative, all right? But I looked at my smartwatch and I was positive. So what did I do? Well, I got on the plane, big mistake, to go to New York City. And in order to go to the meeting, you got to get tested the next morning. And once you know it, I'm bright positive. So I spent a whole week in New York City, all because I listened to my antigen test, not my smartwatch. And so the bottom line is that the smartwatch is more sensitive than an antigen test. So it's not a specific, right? I didn't know this is COVID. And so I got on the plane, that was dumb. But it is more sensitive. So it picks up these anomalies. And that makes sense because doing this continuous monitor. We can get other signals from a smartwatch using machine learning. I won't get then. So these aren't clinical grade measurements, good enough to early alert you that something's off. Here's the infrastructure we built. You can display all this stuff back to your phone at whatever resolution you want, daily, weekly, monthly. Brings in your wearable, omics data, clinical data, all on a smartwatch so you can see this. So this is Mike Snyder's world. I envision a world where people are getting their genome sequenced before they're born. And then together with more deep data measurements from biochemistry and wearables, we'll be able to better, predict risk from the genomes, catch disease early, monitor and treat disease, all using these big data platforms. That's the plan. And so again, I have an amazing lab. I'm going to wrap up. I won't have time, but great microbiome collaborator, George Weinstock, great clinical collaborator, Tracy McLaughlin, great machine learners, and a whole team around wearables and microsampling these days. So we're running all our studies with microsampling. It's just a lot easier.
Mike, great talk. One issue about personal profiling is that most of these are global platforms for research. There will be no widespread adoption unless clinical labs buy in. validate them, and offer them as quote unquote tests. What do you think about that, Mike?
Yeah, no, that's spot on. And our mission, believe it or not, is to take it to that level. So this was a research study I presented, shows it works, got the platform working. Same with the wearables, the microsampling, but we are spinning this stuff off. So YOLO actually will give you they'll run the test in a CLIA lab. They'll give you an absolute value of metabolite concentrations, which have actionable information associated with it, with these metabolites. So we can't make medical recommendations. We're not at that level. But we can make show you levels of certain markers, just like a smartwatch can. So we can give you measurement information, and the goal will be to get FDA approved tests. That's going to take time, and you're spot on, Akhilesh, as usual. So we've got to get that all set up and implemented. But this was the first step, showing it. We spent 6 years working on this microsampling, so I think we're ready to start moving this forward.
One of the major challenges of making such multi-omic personalized health markers available routinely to patients will be health insurance coverage for such tests. Will such tests cost over $2,000 per profile? And would insurance payers be willing to cover the costs?
Yeah, great question. That is the number one problem. Who pays? Who pays? Nobody pays to keep you healthy. And to be honest, this is where the US health system is totally misaligned, right? As the head of the hospital used to tell me, Mike, I only get paid when people walk in. They're not going to walk in when they're healthy. So our whole system is set up as sick care. And that, as I mentioned at the beginning, it's broken. So we really need to incentivize people. I think we're going to have to get employers brought in, but I know we'll get it cheaper in the future. We're working on ways of really trying to drive the cost way down so people will pick it up, and you could actually incentivize them to do it. I would argue in certain areas, there's been some modeling around this for cardiovascular disease. You can actually make a pretty good argument that people at risk should do this, because if you can, like I mentioned before, the heart case, what we call early, that personal drug, if they wind up in long-term health care or some problem because they had a heart attack, that's super, super expensive, or the ICU. So the point is that certain areas, it's very easy to make an economic argument for this. Other areas, it's hard to from the payer standpoint. But I think we'll get this to a point where, as we emphasize healthcare and prevent things, that people want to join these prevention. And I'm a believer you should get a discount if you wear a smartwatch. I mean, the smartwatches, first of all, aren't cheap. They probably should come with a health plan. And yeah, if you get your genome sequence, I think you should get a discount. So anyway, we can talk about this for hours, but you hit a spot on problem there.
I remember your talk in Rochester at the Individualized Medicine Conference in 2012. You had talked about how you discovered you had transient diabetes associated with an illness, but it looks like now it was more of a precursor/warning. Any progress on tools to distinguish transient dysregulation versus warning of future condition?
Why I'm a believer, I never assumed it was transient. I just got it under control by a lifestyle change. And I know that blip looked short, but that was a year of I was up for a year. It wasn't like a viral infection. If you get a viral infection, most people have glucose go off for a few days. That's common, but it doesn't stay up for a month. That is uncommon. So I had to do an intervention. In my case, I did get it under control with lifestyle. But I'm a believer, everyone, and you may know gestational diabetes is almost a forerunner of full-blown diabetes later in life. So these things, and these folks we're seeing who show these sort of transient, you call it transient deviations in their glucose, so they're on their way, in my opinion, and we see evidence of that to becoming full-blown diabetics, just like the gestational diabetic folks with that. So I do think it is a forewarner, and I didn't show our glucose monitoring, but we're doing a ton in that area, continuous glucose monitoring. And I've spun a company there that's actually taking it to a whole new level that can actually, make predictions on people's diabetes and foods they should eat to try to be able to manage that better. So we need a lot more activity in that area, in my opinion, as a medical establishment. I think the diabetic endemic is worse than the COVID pandemic.
What is the false positive rate in case of increased baseline resting heart rate? Knowing that the RHR, or resting heart rate, that knowing that RHR has increased triggers some kind of psychosomatic illness.
Yeah, all right. Well, I don't think they're false positives. What they are is they are stress signals, we are saying, and they're not false. They're just not COVID. And the number one source, we ask people to annotate these red alerts. It is workplace stress. So in a sense, the mental stress from work is actually probably the top. But the flip side of what you're asking, and I get asked this a lot, aren't you going to turn people into hypochondriacs by getting this, not just the wearable data, but other data in general back to them? And my own view is I don't think so. It's like you don't drive a car around without a dashboard because you're worried it's going to break down and things. You kind of want to know. Nobody wants their engine light to go off, but you do need to have a check engine light to know something's going off. And I think you learn how to deal with that. And that certainly the people in our cohort, they're all very pleased to be in their site. Now, there might be some people who would freak out, and maybe the information that goes back to them should be better managed. But I think most people can handle this. This is kind of alerts that you're getting, and it doesn't mean for sure you have this, but it's things that do trigger. It means something's off. And if it's, we did see someone off with the liver enzyme, and he actually just changed his diet and got it under control. It was way off. I mean, it was out of normal range. And that was just really valuable and fixed with the simple lifestyle thing. So I think most people find this super valuable.
With wearables, was the data collected from the wearables and uploaded to a database using an application programming interface? Also, did you encounter any issues with device calibration, syncing to the device, and/or troubleshooting? Who generally handled all the tech support?
Yeah, good question. There's a ton in there. So we do have our own API with my company. For the others, we have to collect it through their API and get it transferred over. It's a little complicated, but we do have a big team around this. Trying to remember what else was in there. Too bad that got deleted. Let's see. So we do have a team around this. There's a whole data cleaning operation on this. What else was in that question? Do you remember?
Who generally handled the tech support? For example, that is the wearable device company, or was it your study team?
No, it was sometimes it's them. It's mostly us. You do need a coordinator who's all on top of it, because a lot of people, they don't sign up quite right, or they aren't charging their watch, so they got to be pinged. We have to remind people to keep their watches charged. The sign up, because we're super secure, I didn't get into this. Everything's encrypted. We have these keys they have to paste in. It's not as simple as just downloading your app for a watch. You do have to paste in this. It's simple, but a lot of people that we're trying to serve, they're maybe not quite as comfortable with apps and this sort of thing. So you better have a dedicated coordinator to any wearable study you do.
Yeah, definitely.
Great question.
How many people, or the N, do you need to feel comfortable about clinical value of this platform, aside from N of 1, personal monitoring or personal patterns? Basically, how many people do you need?
Yeah, well, I am a big N of 1 guy because I think we're all different. I can always detect when something shifted in an individual person, because there are these anomaly detections. You look for shift from your baseline. So what do you need? I don't know. That's something we're going to be working out with the FDA and a lot of what we do, and see what they'll accept. There's this general idea, especially reviewers have this idea. If you don't have 1,000 people, it doesn't count. I think that's true if you're trying to see differences between people and how they might respond differently, but you can tell a lot, again, just from these anomaly shifts. And you do have to build these baselines. Every baseline is different. Every baseline is personal. And so you have to use people's individual baseline to look for that shift. I think the principle will hold it even after a small number of people. But what the FDA will approve, I don't know. We'll have to figure out what the heterogeneity in the population is. I mentioned before, we only found 80%. We missed 20%. People get COVID. And that's because we have a hard time getting a stable baseline on those 20%. I think with more data and the right data, we can do better.
Yeah. I was wondering if you use air quality index as one of the variables in your studies. I came across some studies that the daily AQI, or again, air quality index of a location, city, where an individual is located could affect their productivity and health. And I guess this is kind of going into the exposome.
Yeah, we have a whole separate study. I didn't put that in here around the exposome, and I have an airborne exposometer Where did I leave it? It's always right next to me. I'm kind of, I left it in the other room, which is terrible. But anyway, it's a small airborne exposometer, measures biologicals and chemicals you're exposed to, which is a paper out this summer. Go ahead and look for it. I'll try and put it in the chat. But we can tell, start correlating, external exposures, both biologicals and chemicals, with your internal metabolome and microbiome and things. And as you might imagine, there's a lot of correlations between inflammation, even glucose on your inside, with external exposures. This is a very underexplored area. I think it's an area huge, really ripe for opportunity. So I'd like to see the world spend a lot more time on this. It's something we can control too, right? You can control your external exposures.
I agree as well. A lot of, as we know, like carcinogens are everywhere, pesticides are everywhere. We got to be able to understand how.
You got it. And they're 100% everywhere, but their amounts do vary from one location to another.
What kind of statistics are you using for the AI related laggard determination that you showed in 2 of your slides?
Yeah, I think the best thing to do is go look up. We put that algorithms up on the hub, and they look for. In general, we're looking for things a certain number of standard deviations away, but we do set up different windows for that time delayed lag, and we test different windows and settle on one we like, I guess. Although we varied it for different parameters.
You mentioned variances of personal baselines of profiling. How can we distinguish whether a personal baseline level skewed from average is within the normal range or reflects an inherited abnormal state?
You mean skewed from average, from their average? Skewed.
I think average of the entire population you're measuring.
Yeah, well, you hit the nail on the head. That's the thing. There is no real average when it comes to health. I think you really have to see your own personal shift.
Do you control for gender or analyze for gender sex-specific differences? She's thinking about menstrual cycle effects on resting heart rate and perhaps other parameters.
Yeah, another great question. So the answer is obviously it's built in from the personal baseline, whether you're male or female, but the menstrual cycle is something we do correct for. I didn't get into this, but people have different behaviors on weekdays versus weekends. Just as an example, your heart rate and your temperature change, women's temperature change, depending where they are in the menstrual cycle. So the answer is yes, you do have to correct for it. I don't think we were perfectly corrected when we launched our first algorithms, but it's still good enough because the shifts you see are pretty good. But nonetheless, now we're even more sophisticated. And for most things that is put in. Not when we have data, we put that correction in. Great question though.
You mentioned all this data that's being tracked with wearables and multi-omics profiles. And you also, were generous to share some of your personal stories as well with how you got through the type 2 diabetes and the Lyme disease and all. But what I'm curious is, how do we make this data, like, okay, right now it's 2023, but let's say 25 years from now, 50 years from now, how do you imagine the future is going to be how we make all this data more actionable? I mean, yes, we know we should stay away from sugary foods. We should, exercise, lift weights, moderate stress levels. But we don't need multi-omics data to tell us that. But what's the power of all this data dense, these, as Leroy Hood likes to put it, these dense dynamic data clouds?
Right.
How is that going to, let's say, prevent disease? Or actually, if you have a disease, how are we going to use multi-omics data to shift our omics profiles to something to actually cure the disease or mitigate symptoms?
Yeah, I think for chronic diseases and things, you're hitting the nail on the head. We need behavioral modifications. I do think continuous feedback, like I say, if you saw the continuous glucose monitoring, it really does give you the kind of feedback you need to modify your behavior. You can't, that information is so powerful. So that's a real-time one. That's an easy one. Other things like, you're supposed to exercise more, lose weight. Well, yeah, we need motivational programs for being able to do that. But I still think, infectious disease is another easy one. That one's easy to spot, right? When you getting alerts for illness, you take action by sitting at home. Don't go out and infect everybody else. So some stuff's easy. Some stuff's going to take a lot more work. I think the more we can make it easy to do work, I think the more we can realize, like for this microsampling, just knowing what foods trigger what could really, like inflammation or suppressing inflammation, you can use that in everyday life. So I think they can be teaching tools that will help you modify behavior. We still need more to do to go along with that.
Do you think these omics profiles could suggest certain drugs you should take as a prophylactic, or even vitamins and supplements?
100%. I think the diabetes is a good example. There are many subtypes of diabetes. I mentioned I don't release from the pancreas. There are other people with beta incretin effects, others with muscle insulin resistance. And different drugs can actually mitigate those effects differently. So I do think you could tailor drugs much, much better. You can also see response better. Imagine you take a drug and then right away at home do microsampling, see if it's working in 3 days rather than waiting 3 months. So I think we can incorporate these technologies to manage people's health much better in real time and with a better effectiveness. All right.
Thank you so much, Dr. Snyder. See you next time.